अब हम बात करेंगे प्रोसीजर की दैट कैन बी used or that can be performed to get the results from microarray sample. We are going to talk about two different samples so that we can see the difference between the gene expression under two different conditions. One sample is from the cancerous human skin tissue. The second one is from the healthy human skin tissue. सबसे पहले हमें आरएनए एक्सट्रैक्ट करना है. आरएनए एक्सट्रैक्शन के लिए वी कैन यूज द रेडी टू यूज किट्स using the column, or we can use the solvent method that includes the phenol chloroform method. And this RNA which is isolated is basically total RNA. This total RNA contains ribosomal RNA, messenger RNA, transfer RNA and other types of RNA which are present in the cell. In order to get the precise results, it is better to purify the messenger RNA, and for that purpose we can do the column chromatography. जैसा कि हम पहले इस पे बात कर चुके हैं, column chromatography में we have certain beads which are labeled with poly T. Messenger RNA has a poly A tail. उस purification step में जब हम जाते हैं, then poly A tail binds with the poly T and all the other RNAs are washed off. Later on, the messenger RNA or poly A tail RNA, they are eluted from the column in absolutely pure form. Once we get the poly A RNA or messenger RNA, reverse transcription is done. For reverse transcription, we use oligo DT primers, reverse transcriptase enzyme. Most of the time we use MMLV reverse transcriptase and fluorescent dyes which are going to label the cDNA. Cyanin 3 is going to be added to the healthy cells and cyanin 5 is going to be added on the cancerous cells. ये चीज़ यहाँ डिटरमिन करना बहुत ज़रूरी है कि आपके कौन से सैंपल का जो कलर्ड cDNA है वो कौन सी फ्लोरेसेंस एमिट करेगा, so that at the end we should know that green color is from the messenger RNAs coming from healthy tissue or from an infected sample, and red color is coming from which type of sample, type 1, type 2, जौन सा भी आप उसको nomenclature दें, healthy, diseased, controlled, experimental, sample number 1, sample number 2, whatever is the labeling. Using this method we are going to get first strand cDNA synthesis. The first strand cDNA is required because we are going to hybridize this single stranded cDNA with our probe or with our target DNAs on the microarray. Once the cDNA is ready, we have microarray, and that microarray is going to be assembled in the microarray chamber made up of stainless steel, and the sport slide which has a gasket is going to get our this cDNA sample, which is going to be at the base. Microarray is going to be at the top in the upside down position, so that the cDNA is going to get fully exposure to the microarray. अब इसमें हमें एक चीज़ इंश्योर करनी है that the chamber should not be opened, the cDNA should not be exposed to air. उसकी रीज़न क्या है कि हमने जो cDNA यूज़ करना है उसकी वॉल्यूम बहुत कम होगी. अगर हम उसको प्रॉपरली कवर नहीं करते स्पोर्ट स्लाइड के साथ, तो विद इन अ मिनट जो लिक्विड पोर्शन है दैट इज़ गोइंग टू बी इन एयर evaporated because of temperature, and then we will have problem in hybridization. Once this single strand DNA is going to hybridize with the cDNA, after the hybridization period, we are going to wash it, and then we are going to use the scanner for fluorescent detection. अब यहाँ पे एक चीज़ अगर कंपेयर की जाए, हमने जब नॉर्दर्न ब्लॉटिंग के हवाले से बात की, सदर्न ब्लॉटिंग के हवाले से बात की, या वेस्टर्न ब्लॉटिंग के हवाले से बात की, wherever we wanted that one molecule hybridize with the other molecule. There we followed a hybridization step, and after the hybridization step we followed the washing step. Washing is mandatory so that all unbound or unused material should be washed off, and whatever is left behind is going to give you the true picture of the results. This microchip or microarray is going to be inserted into the scanner, and the scanner is going to reveal the fluorescence which is coming out from the spots in the form of green light or in the form of red light. And if particular spot hybridized with both the cDNA samples, then green and red color is going to give a yellow colored appearance, which is quite much visible here in this picture. The green color reflects sample number 1, red color reflects sample number 2, and yellow color reflects the messenger RNA or the gene expressed in both the samples. In this way, using this method, in relatively shorter time, we can screen a large number of genes, and from these preliminary results we can go further, we can move further that which gene we should focus on, and further we can make a network of the genes which are interacting with each other during any particular condition.